Imagine taking a massive dataset of human genetic markers, thousands of individual sequences of DNA, and plotting them on a blank canvas. This dataset contains no geographic labels. There are no tags for French or German. It is raw, unlabeled biological data. This chart shows the result of running that exact data through a strictly mathematical algorithm. The algorithm organized those unlabeled genetic markers into an almost perfect geographic map of Europe. This result highlights the core challenge of unsupervised machine learning. In the absence of labels, we must find a way to extract a meaningful signal buried within raw, high-dimensional noise. The specific algorithm responsible for revealing this geographic structure is called Principal Component Analysis, or PCA. It is a linear transformation technique used to simplify the complexity of large datasets. To discover the true underlying structure of any dataset, the algorithm must strip away redundant noise and find the data's most essential dimensions. High-dimensional complexity is often an illusion caused by correlated attributes. Consider a theoretical dataset tracking the maximum speeds of various motorcycles. If we record speed in both miles per hour and kilometers per hour, these two variables tell the exact same story. Because they are perfectly correlated, the data naturally collapses into a one-dimensional line. The second dimension provides zero additional information. This redundancy appears throughout real-world biology. In the iris flower dataset, a flower's petal length and its petal width are similarly correlated. They grow in proportion to one another. This reveals the intrinsic dimensionality of a dataset, the true lower number of independent variables required to describe the data accurately. PCA automatically identifies this intrinsic dimensionality by finding the natural axes of the data and discarding the redundant dimensions. PCA assumes that high-dimensional data points naturally live close to a lower-dimensional linear subspace. We define a formal linear transformation from our original high-dimensional space, X, to a compressed latent space, Z. This transformation is defined by a parameter matrix, W. The process begins with encoding. We take an original high-dimensional input vector, X, and multiply it by the transposed matrix W to compute its latent coordinate, Z. The decoding step allows us to reverse the process. By multiplying the latent coordinate Z by the matrix W, we reconstruct an approximation of the original high-dimensional data, known as X tilde. The central problem is finding the matrix W that defines the optimal lower-dimensional subspace. Finding this optimal matrix can be achieved through two philosophies that arrive at the same answer. The first philosophy treats PCA as a geometric approximation challenge, where we must minimize data loss. This visualization shows a three-dimensional cloud of black data points being intersected by a translucent two-dimensional plane. This plane is the lower-dimensional subspace we are trying to optimize. As shown by the red orthogonal lines in this plot, the optimal plane is the one that minimizes the physical distance between the true 3D points and their compressed locations on the 2D surface. We define this physical distance mathematically as the reconstruction error. The formal objective function, J1 of W, minimizes this error by calculating the sum of squared L2 norms for every point in the dataset. In this equation, X represents the true point floating in space, and X tilde is the exact location where it's landing on the plane. Minimizing the reconstruction error ensures we select the subspace that destroys the least amount of spatial truth when the data is compressed. The second philosophy focuses on the utility of the data points after they have been projected onto the lower dimensional plane. A projection is useful only if it preserves the distinctiveness of the individual data points. This diagram shows a failed projection. The axis chosen cuts across the natural shape of the data, forcing the points into a tight cluster near the origin. This tight clustering destroys the signal, making previously distinct data points indistinguishable from one another. Successful projections align with the longest natural spread of the data. Here, the blue size dimension preserves the distribution, while the orange other dimension collapses it. We call this spread variance. It effectively measures how widely distributed the data points are along the projected basis vectors. By maximizing variance, the algorithm guarantees it preserves the most distinguishing characteristics of the original dataset. PCA relies on a specific geometric theorem. Minimizing reconstruction error and maximizing variance are identical operations. Consider a single data point and a candidate projection axis. A right triangle connects the origin, the fixed point, and its projection. The orthogonal side is reconstruction error, and the adjacent side is variance. By the Pythagorean theorem, decreasing error proportionally increases variance. Solving for the tightest geometric fit automatically yields the most informational spread, unifying the two mathematical philosophies into a single algorithm. Maximizing variance allows us to extract structure from raw, unlabeled images. These clusters represent handwritten digits organized by their natural axes of variation. The algorithm separated the 2s from the 6s without any prior knowledge of what these numbers represent. This brings us back to the DNA map from the beginning of the video. The x and y coordinates on that map are simply the two axes of highest variance in human genetics. By simply minimizing mathematical error, PCA serves as a foundational lens for machine learning, revealing the hidden natural order inside massive, chaotic systems.